Lastly, I'll give a short sort of intro to sort of how at Silicon Therapeutics we were thinking about integrating biophysics and how it helped to study the conformations of a particular protein of interest and generate models that can be used to drive drug discovery programs. Okay, so for the first story, I'll give a brief sort of blinded target introduction to the project that I co-led. So this was an early stage immune oncology program. It was a really exciting program. It was identified through CRISPR screening. So knockdown in CRISPR suggested that it inhibited cancer cell growth and tumorigenesis. And this result from the CRISPR screening was validated against multiple cell lines that it did indeed inhibit tumor cell growth and activated type 1 interferon response. The target protein is a nucleic acid binding protein whose function involves binding and altering of nucleic acid. The loss of this protein is beneficial and it overcomes common mechanisms of resistance immunotherapy drugs. It's a first-in-class target with no precedence for any type of drug discovery. So it's a novel target, and so when we think about the drug that we're trying to design, it would be a first-in-class small molecule inhibitor or a PROTEC degrader that induces the same type of interferon response that was observed in the CRISPR cells in knockdown experiments. So to begin, there were several challenges that hampered an effective drug discovery campaign. First and foremost, the target was extremely challenging, both from a structural biology and enzymology perspective. So the target had no published crystal structures, which limited the use of structural-based drug design, and there was limited understanding of enzyme regulation. The ligandability was also very difficult in terms of this being a nucleic acid protein whose binding site is attuned to binding charged nucleic acids. So it's categorized as a sort of hard to drug target class in general. And there was no tool compounds or any binders outside of nucleic acid that could be used for assay development to guide either binding or functional screens. And to combat that, or to compound that, there was no validated assays established from the perspective of binding or plate-based biochemical assay to test for functional inhibition. So we had to sort of develop each and every one of these techniques in order to have an effective drug discovery campaign. So we started off with taking on the first challenge, which is to generate a, Sorry about that. So we started off with the first challenge to generate a high-resolution crystal structure. This was kind of a large protein engineering campaign that was guided by HDX-MS that was collected at UMass. And so the idea was we would use the HDX and the molecular dynamics simulations to understand sort of the dynamics of the protein, and then use that information to guide protein engineering. So the workflow that we utilized started off with identifying optimal domain boundaries for the protein constructs to ensure that we're getting high yield of the protein and the protein was stable and can be expressed at high levels. Then we leveraged the HDX-MS and molecular dynamics information to guide sort of a construct engineering using techniques like surface entropy reduction, loop alterations, sort of this alpha tag nanobody, which is essentially utilizing a nanobody that facilitates crystallization and tag that and bind that to the protein of interest. And again, these sort of computationally informed alterations guided by the HDX. So from this workflow, we generated over 100 protein constructs, and which resulted in two mutated forms of the protein that were engineered that yielded very robust crystal systems, which is sort of shown here with the nice crystals that we see on the tray here. And we were able to get a very high resolution crystal structure. It's a novel crystal structure, and we were able to use it routinely for drug discovery. So now we have the crystallography in hand. We then moved to transition into a hit finding approach. We took 3 different hit finding strategies to generate chemical matter for this novel target. The first was the fragment-based drug discovery, which I performed the NMR fragment methods myself. So we did two fragment-based screenings, F19 NMR, which I'll show a little data on using our in-house fragment library. These experiments were performed with the 600 at UMass. And we also complemented that with a fragment screening by crystallography approach, leveraging the novel crystal structure I just described. We also used two other screening methods, DNA-encoded libraries, using commercial kits and a covalent screen, which was actually a functional readout in cells, which used our internal covalent library to react mostly with cysteine residues on the protein that could potentially lead to inhibition. The most advanced hit finding was the fragment-based screening, so I'll mostly be focusing on that for the next few slides. So I do want to give a little shout out to the NMR work that I did at UMass. So Royvan had an internal fragment, F19 fragment library of 1,500 fragments. Fluorine-19 has a number of advantages, including high sensitivity, large chemical shift dispersion, and favorable relaxation mechanisms. So the small molecules are very sensitive to binding when they interact with large proteins. So the sort of result of that, set up a fragment screening campaign using relatively low concentrations of protein, tens of micromolar, and also relatively low concentrations of compound. Here I only use 50 micromolar. And the nice thing about fluorine NMR is that when you define your fluorine fragment library, each fragment has one peak in a fluorine spectrum shown here. And you're able to easily correlate each signal in this spectrum back to the fragment molecule. This screen can be, and because of that, you're able to screen in pools. Here I did a fragment pool of 30 molecules. And you can see the sort of, this red on top would be the reference, when the hit molecule is shown here, which is the loss of signal in the T2 CVMG experiment. These experiments were done on the UMass 600. They utilized this Burbop excitation pulse and refocusing pulses, which are really nice and allowed me to pool both the CF3 containing fragments as well as the CF and the C aliphatic ones into a single sample and collect the data in one experiment. Whereas you would typically need to do two experiments if you were using hard pulses to collect the CF3 region separately from the CF. So in total, screening 1,500 fragments occurred in only 3 days of acquisition time, and we identified 40 hits, or a 2.6% hit rate from this screening. The next screening that we did was a fragment screening by crystallography. We leveraged our in-house crystal system. It was highly robust, consistently produced crystal structures of less than or equal to 1.5 angstroms, and nicely it tolerated 30% DMSO. So in a fragment screening by crystallography, you're soaking fragments at high concentration. So if your stock is 500 millimolar compound, 30% DMSO is 150 millimolar compound soaking into the crystal. So that really gives us a good opportunity to get a lot of hits. We screened over 1,000 fragments, including the F19 hits that we derived from the F19 screening. And we ended up with about a 10% hit rate. And so the sort of workflow what we did was we screened both internally and through a vendor that's expertise in high throughput crystal screening. We developed and utilized refinement and ligand identification workflows so that we can process all of the crystal data that we were getting in an efficient manner. And then you can see the sort of result that you get here with the compound in green. You can see the really nice electron density with the pi cloud hole, which is something that you get, you observe when you have a really nice crystal system that produces, close to 1 angstrom resolution structures. So because we had so many fragment hits by crystallography, the question became, how do we prioritize the hits for optimization? And so this table on the left shows how many fragment hits we got, as well as all of the sites that fragment hits were observed at. Uh, Unfortunately, none of the fragments were found in the active site of the protein, which is not surprising considering the active site is attuned to binding nucleic acids. So the question is, how do we prioritize which fragments for optimization? And would there be a potential for an allosteric inhibitor, or would this be just a binder that we can leverage for targeted protein degradation? So our thinking was, obviously we want the hits that we prosecute to be structurally enabled, which by virtue of the screening method, they were. But we really wanted to focus on our orthogonal binding validation, which we primarily leveraged NMR. But we also want to leverage our computational platform to assess the binding pocket, especially for progressibility and growing out these fragments. So all of these sort of features were required for the selection and optimization of the fragment. So I'll give a quick intro to how we thought about using predictive computational sciences to execute the pockets of interest. So we used a combination of methods, the first being mixed solvent MD. So this is a molecular dynamics simulation where many of the water atoms that are solvating the protein are replaced by these small probe-like molecules. And these molecules essentially pepper the binding sites of the proteins. And if they stick for a considerable period of time, it's considered a hot spot. And so you can use these interactions that these small probe molecules make with the proteins to construct a sort of pharmacophore and binding site map. And so obviously we would hope to see them correlate with the fragment pocket that was identified by screening, but also to look for sub-pockets for areas for growing out the fragments. Another method we used was GIST and F-pocket analysis. These are, sort of a binding site prediction that is sort of learned from success in other binding sites for drug targets, and so it understands those features, as well as the GIST, which is a water map method, which essentially looks for high energy water. It could tell us, if we grow the fragment in one direction and displace those high energy waters, we should see an improved binding affinity for the grown fragment. The other two methods are other molecular dynamic simulations. One is the RBFE method, which stands for relative binding free energy. This is a very advanced sort of in silico binding method. So what it does is that it computes the sort of relative binding free energy of a given molecule as you sort of scan out the different growth vectors. So if you add building blocks to, R1, R2, R3 vectors, you should see a decrease, more negative free energy. And that suggests, one, that there's room to grow, but also that there's, what direction to grow and what are the kind of interactions that can be made and are predicted to be more favorable. One of the caveats here was that the fragments are quite small and we're not making enough interactions. And so we're not stable in the binding site. So we leveraged another experiment, which was either plain MD, just to make sure that the fragments live in the binding site for a significant amount of time and make the kind of contacts, as well as a random accelerated MD, which is just to add a randomly accelerated force to push the fragments off to just get a sense of whether their interactions are truly stable or not. So we performed these analyses to try to figure out which is the best pocket to proceed. And we also performed the experimental hit validation. So with the two screening methods for the fragment screening, the F19 and the crystallography, obviously the F19's hits were soaked into our crystal system to get co-crystal structures, and the fragment hits from crystallography were tested in NMR, usually ligand observed at STD NMR. We also generated C13 methyl labeled protein in insect cells, which unfortunately I don't have the slides or any data from this, but this was quite an effort to just prepare the protein in insect cells using isotopically labeled amino acids, which provided sort of the most confidence binding assay that we can use to validate these fragment hits, which were fairly weak, but the protein observed NMR, we still would see some shifts for some of the fragments, and became our most high confidence assay. And as a result of this, as well as the in silico evaluation, we ended up with two sites, site 3 and site 4, and we deemed site 4 the highest priority. As you can see from the co-crystal structures here, so this is the apo state. There are two arginines in this site, and the fragments induces a sort of conformational change in one of the arginines and is making really nice contact with these two groups. And so we thought that that was sort of a key interaction that we can build off of. But also we saw many of the hits that were in site 4 from the crystal screening showed binding by protein observed NMR, and many of those hits were from the F19 fragment screen. We also thought that there was a potential for an allosteric type of inhibition mechanism because we saw some perturbations in HDXMS when the nucleic acid bind the protein to this site. So we thought maybe there was some dynamics that we could exploit for an allosteric pocket. Site 3 was a lower priority site. It had an actionable sub-pocket that we can potentially grow our link fragments into, but not many of them conferred by protein observed NMR. Now that we have the fragment starting points, we embarked on our sort of research operating plan to prosecute the fragments. We wanted to take those starting points and use our computational binding predictions to determine, how to grow the fragments and to influence the sort of med-chem decisions there. Also, we wanted each of the fragments we started with to be structurally enabled, and for every round to try to structurally enable subsequent rounds of fragment enumerations. And many of these assays from the biophysics side, including SPR and protein NMR, were our preferred methods for prosecuting these fragments, but both of these biophysical methods had challenges. So from the perspective of SPR, we would have liked to run SPR, which is sort of a high throughput binding method, but considering that our fragment starting points were somewhat insoluble and the KD was too close to the solubility limit, we couldn't routinely run this SPR assay. In addition, for the protein observed, although we used this to validate the fragment hits from screening, there was issues in generating sufficient amounts of labeled protein, especially from insect cells, so we couldn't leverage this method routinely. And even more, the sort of biochemical assays we didn't think was appropriate. Although we did develop a novel assay, we didn't think it was appropriate for fragments until the affinity reaches a certain threshold. So what we ended up doing was pivoting to a F19 competition assay, since many of the hits were derived from F19 screening and they validated by crystallography. So we have an understanding of the site, and we know we want our subsequent fragments should be competitive, since they're all designed to bind to that particular site. So we took the sort of each SAR cycle set of compounds and prosecuted them by the F19 binding assay, which we expect to show rank order of the new molecules, and then we emphasized sort of orthogonal validation, which usually was protein observed NMR. But eventually we wanted to establish an SPR assay. So really quickly, an overview of the F19 competition assay. So I really like this assay. This is a figure from Aztecs showing that NMR reporter assays are really useful for bridging the gap between the weak sort of millimolar fragments and the sort of more potent, hundreds to tens of micromolar, enumerated ligands that can be more easily relied on in SPR and biochemical assays. And so it works just like any other FP competition assay. You have the free ligand, which gives a really strong signal in the absence of protein. There's a reduction in the signal intensity upon binding to the protein of interest. And then you add in your competitor molecule, and you should see competition. And so the sort of spy molecule or probe molecule you're using should go back from the bound state to the more free state when you add in the competitor ligand here. And of course, you could do it in titration mode and get Ki values. And so the expectation again was that the level of displacement that the competitor molecules have will rank order correctly with Ki and should correlate with the Kd. And so we developed this sort of strategy then from the medchem side to advance these fragments at site 4 with the goal of scoping out the pharmacophore and increasing the size and ultimately increasing the ligand occupancy and the number of interactions the ligands makes to improve the overall potency. So we had 3 approaches that we took initially. The first was an SAR by catalog, or we did 2 SAR by catalog approaches. One was a pharmacophore fitting, and the other was a pocket shape, which basically took the sort of features of the fragment hits that we observed and looked commercially for other molecules that had that same feature, as well as leveraging both the shape and the pharmacophore feature protein side of the pocket shape. And this confirmed, we reproducibly saw binding of this pharmacophore, and by crystallography, we saw that they engaged both arginines. Another interesting method we took was fragment merging. This leveraged our crystal structures of our fragment hits, and we saw that 2 fragments were spatially overlapped in the binding pocket. And so we generated this merged fragment, which became the scaffold that we tried to advance at the highest priority. So this is sort of a blinded view of this. And so a little bit more details on that. So this is the F19 hit that we obtained from the F19 screening, as well as the crystal hit in the sort of blinded form. Both were structurally enabled, and again, they merged, in space in the crystal structures very nicely. So it was really quite amazing when we made this merged scaffold, how well the merged fragment aligned to spatially to the original parent hits. And so this is the sort of compound we used and took forward with our sort of enumeration workflow. So we took this scaffold. It was not as potent as we hoped. So we needed to grow and expand the sort of interactions that it was making with the protein. So the approach that we took was a chem approach using what's known as a reaction-based enumeration. Essentially, what we did was we synthesized an intermediate of this scaffold, and then we took synthetically compatible building blocks that enabled sort of a one-step, very easy chemistry synthesis, so we can make a lot of molecules very quickly and at defined vectors. So for example, the R1 vector, we can link all of these molecules here, as well as at different vectors, throughout the scaffold. And because this approach can quickly, sort of build up a large library of molecules, we wanted to make the process more efficient by using the computational platform. So we virtually enumerated all of these molecules, growing from the building blocks at each vector, and then we applied a simple scoring and filtering process to only synthesize the compounds that are expected to based on the predicted potencies. And so these are the sort of numbers that of compounds that we were able to, or we were in the process of making. Unfortunately, that's when this sort of project sort of ended, and Roivant decided to change directions. But I will say that this project is still ongoing at Roivant in some capacity. It was partnered with a large pharma, and so potentially that's still some active work on this target that's utilizing some of the activities that so we sort of set the stage for. And so really the story is a collaborative effort to sort of remove the roadblocks for effective drug discovery. So we have a new crystal system that has very high resolution. We're able to deliver many co-crystal structures. We have starting points for fragments that have been validated by NMR methodologies, as well as derived from NMR methodologies. We have additional hit finding and covalent screens. At the time that I was there, we're still ongoing. And we also have developed SAR driving assays, the NMR competition, as well as some early work in SPR that we could leverage for a successful project. If there's no questions on this project, I'll move to the next story, which is the diatomite project. So at Roivant, we were interested in targeted protein degradation. We were interested in that also for the previous project as well. So just a quick introduction to targeted protein degradation. You can think of sort of traditional drug discovery targets as inhibiting or functionally regulating protein activities such as enzyme inhibition. But targeted protein degradation is distinct from that in the sense that it opens up sort of unlocked targets that you don't need to inhibit enzymes. You just need to identify binders. And then the way it works is it leverages sort of protein degradation, the ubiquitin proteasome machinery within the cells. So this is, for example, you have an E3 ligase with an E3 ligase binder. You have a protein of interest as well as the protein of interest binder, and then you form what is known as a protein degrader molecule. And then in the second step, you form what is known as a ternary complex linking the POI to the E3 ligase, which then transfers ubiquitin onto lysines. Once the ubiquitination occurs, the complex disassembles and the protein is degraded. And hopefully you see the sort of pharmacology that you're interested in. So with this approach comes a number of challenges. So I tried to exemplify those here. So the example that we'll use in subsequent slides is the SMARCA2, which is a protein of interest for oncology. And so this is the ligand for SMARCA2 that's been developed. And when you think about degrader synthesis, you think about what the exit vectors are. And so you think about how to, where there's solvent accessible channels so you can grow a linker molecule and connect it to, for example, VHL, which is the E3 binder. And the structure of its warhead is shown here. And so you can imagine multiple exit vectors off of this protein. Only one exit vector is shown for SMARCA, but you can imagine multiple exit vectors that are possible that are pointing out the solvent. And so it's not usually obvious which one is the best, so that the usual approach is to generate libraries of different linker molecules and connect them to each exit vector point and then assay them, which is sort of a very empirical process. And so this is the structure of one of those molecules shown here. And it's known through experience that slight alterations in the structure of this molecule can influence the sort of ternary complex, and that could influence the subsequent degradation. And it's usually not really well understood how these two correlate and how the changes in the linker can influence the ternary structure. And to sort of build on that, it's sort of widely appreciated that ternary complexes are dynamic ensembles. So In this example, I'm showing a crystal structure of BTK to CIP. And in this crystal structure, hopefully you can appreciate that there are multiple different conformations of the same complex in one crystal structure. So, this has a different sort of conformation and sort of interface than this one, and this one has a different angle and a bigger interface than the other three, and they're all in the same crystal structure. Suggests is that ternary complexes are very dynamic ensembles, and it becomes very challenging to figure out which of these models is actually sort of bioactive conformation, and moreover, which of these models to use for drug design or linker design or what have you. So the approach of the sort of platform at Roivant was to use computational resources integrated with biophysics to predict ternary complex ensembles at very high accuracy, and to simulate the energetic landscape so we can figure out what is the most useful conformation, which is predicted to lead to degradation, and what sort of model should we and optimal PROTEC should we design, as opposed to going through the process where you're sort of just doing things very empirically and throwing in a library set. So to improve the sort of efficiency of this design process. So to build up tools to understand what makes a degrader, we use the SMARCA VHL system. There are 3 PROTACs that have all been crystallized, and as a ternary complex with SMARCA2 and VHL. The compounds are shown here, highlighting the sort of change in the linker, as well as the maximum degradation observed in cellular assays. So ACBI1 and PROTAC2 induce a greater than 90% Dmax, whereas PROTAC1 is only 65%. So we want to sort of try to understand these sort of tool systems, and we want to integrate biophysics into the simulation to be able to more accurately guide both the MD, but also the dynamics and energetic landscape. So we leverage 2 biophysical techniques. HDX-MS is performed at UMass. It was utilized to identify residues protected from deuterium exchange in the ternary complex, and can be used for constraints in the protein-protein docking, and small angle X-ray scattering coupled to size exclusion chromatography, which provides sort of the global orientation and was used to help validate long timescale simulations. So the paper has been published, but I'll try to sort of summarize it very briefly. So, 3 things really stood out from this work. The first is an HDX guided weighted ensemble simulation. So this is our docking protocol. And so the weighted ensemble is essentially a path-based molecular dynamics simulation that uses the active residues in the HDX, defined as the interface, as a collective variable. And so this is much more efficient at getting to the final conformation than just a plain MD simulation. And so the figure here highlights the level of agreement that these simulations obtained relative to crystal structure. And this figure is showing the correlation of the contacts that were observed for the PROTEC or for different residues, and how that compared with the crystal structure. And so this approach was determined to be much more accurate than just using docking and HDX alone, because it also leverages the dynamics of the system. Then with these models, we also performed a long timescale replica exchange dynamics simulation, and this was really geared at identifying the lowest energy conformation and the lowest energy states. And so the long timescale simulations were plotted in this PCA plot, where you can see, for example, in ACBI1, the local minimum free energy, which is highlighted in cyan, and the crystal structure conformation are actually very similar, meaning that the crystal structure and the simulation agree that that is the lowest energy state. But for PROTAC1 and PROTAC2, this is not the case. There's a different energy well for PROTAC1, and PROTAC2 actually has two energy wells, one of which is the crystal structure, the other of which is a novel state. And that's really shown here, where you can see ACBI1 and PROTAC2 have very similar conformations. They're the most active. But PROTAC2 also has a sort of two different low energy states, one of which is closer to PROTAC1, which is not as an effective degrader molecule. And so from my perspective, it was very important to validate these long timescale simulations that are actually recapitulating experimental data. So we chose to use SACs, size exclusion coupled SACs of the ternary complex. So we form the complex, run it over a size exclusion column, and collect the SACs data that from the peak that corresponds to the ternary complex. And so these figures here are highlighting the sort of agreement that we saw between the between the SACs and the long time scale of molecular dynamics simulation. These figures are showing two different isoforms, which are not really relevant here. What the important point is that we took snapshots of the MD simulation and computed the SACs, the average SACs of that simulation, and compared that to experimental data, which you can see the correlation here is really good. You can also see the sort of probability density of the radius of gyration, which reflects the size of the overall size of the ternary complex. The SACs data is shown with these triangles, and you can see the probability distribution from the simulation is really good agreement with the SACs data, suggesting that they're sort of recapitulating experimental data and are valid simulations to sort of model and leverage. Finally, we saw differences in conformation between the three different degraders. In sort of a last question, do these low energy conformation explain differences in degradation? And so to answer this question, we took the conformations that we produced with the simulations and modeled them onto the colon ring ligase, which is this large protein complex with the ubiquitin shown here that it transfers onto lysines of the SMARCA2. And so in this simulation and model, what we're doing is showing the probability of lysines, the distance between a lysine and the C terminus of the ubiquitin, because there's what's known as a ubiquitination zone. And so if your protein of interest goes within this zone, or conformations of that complex moves into this ubiquitination zone, then the ubiquitin can transfer onto lysines of the protein. So this data showing here shows that ACBI1, the conformation that it forms in the ternary complex, has the greatest probability of lower distances, which could result in the sort of better degradation potential of this degrader over, for example, PROTAC1, whose other conformation results in larger distances between lysines and the zone of ubiquitination. So this sort of workflow and methodology became part of a spin-off company from Roivant called AtomMap, and so they're still leveraging this technology to facilitate both design as well as structural understanding of ternary complexes and modeling those to aid in the design of PROTACs for discovery. Okay, so in the last section, I'll cover sort of my experiences at Silicon Therapeutics, integrating in silico method with biophysics, especially NMR. And I'll introduce another target of interest, which is the protein called STING, which is another high interest immune oncology target that ultimately Silicon Therapeutics developed a small molecule drug for and is in phase 1 clinical trials. Um, but this protein is highly dynamic, and I'm trying to illustrate that here by showing the differences in conformation between the apo and bound to its dinucleotide substrate. So there are inward motions of some helices, a conformational change that results in a beta sheet formation sort of in the central channel, um, which we denoted as like flap stabilization, as well as other conformational changes that are occurring when the target binds to ligands. And so when you have dynamic proteins, it becomes very difficult to use structure-based drug discovery methods, especially when routine crystallography is absent or very difficult, which is usually the case for very dynamic proteins of interest. And so, how do you account for multiple conformations, and how do you know sort of like a priori which conformation your molecule is inducing, and how do you get the best model for which you can use structure-based drug design to advance and generate more potent molecules of interest. So our approach to sort of tackling this problem is, again, to integrate simulations, both NMR as well as small angle X-ray scattering. And the idea is to say that sort of the overall philosophy was that we want to leverage our molecular dynamic simulations, but they can, if it's a dynamic protein and you're starting off in the wrong state or wrong conformation, the simulation will take a really long time to get to the right conformation. And so adding experimental biases from these two methods can make the simulations more efficient and ultimately get us to a better molecule, which we can act on from medicinal chemistry. And so just to further emphasize the issues and sort of the conformations that are accessible to the STING protein, we can see that simply from this sort of C13 methionine methyl spectrum. So there are two methionines in STING. It's a dimeric protein. And there's two methionine groups, one at a loop and one at the helix here, sort of below the compound binding site. So we showed with 4 different compounds that there are multiple conformations that occur. So in the top figure, when compound 1, 2, and 3 are added, there's a shift in the methionine 271 as a result of compound binding. But the loop conformation is altered differently for each of these molecules, for each of these compounds. So for example, compound 3, you see a small chemical shift perturbation, but for the other 2, you see extreme line broadening and loss of peak, indicating sort of dynamics in this area. With the cyclic dinucleotide, which is its native substrate that activates STING, compound 3, as an overlay plot, you see that both the substrate, the conformation of methionine 214 is very similar as the native substrate with compound 3, but the methionine 271 here is in conformational exchange, and so you see the loss of the peak here. And for compound 4, both methionine peaks are invisible, and so you see an entirely different conformation for this ligand. And so all of these ligands functionally are active, so it really just highlights that multiple dynamic conformations can result in activity of this target. So what's our approach to sort of understanding, in a quick and routine manner, what the relevant bioconformation of the protein is? So I leveraged methyltrosy, which is really advantageous for proteins that are large in size, and so methyls have very favorable relaxation properties. There's 3 protons for each methyl group, and so they give really nice high resolution spectra as well as good sensitivity, and they're applicable for really large proteins up to 100 kilodaltons. So as long as they express in E. coli. And so this is a C13 isoleucine, leucine, valine, methionine, C13 methyltrosine spectrum. And this figure here shows sort of the distribution of the methyl probes throughout the sting protein that we can use to sort of characterize any changes. And so as with any NMR spectrum, it's important to assign each peaks to the methyl groups of the protein. So we leverage an automated methyl assignment using through space methyl-methyl NOEs, which I'll talk about on the next slide. And then the workflow would be to titrate the ligands in to see the conformational changes, and you hope to see, either patterns form with different chemical series, or we would utilize the sort of methyl distance restraints as well as C13 edited noses to give sort of the sparse intermolecular NOEs between methyls and the ligand of interest, and then incorporate these into simulations to drive the simulations into the most active conformation. So the idea is that we're not solving a de novo structure every time, but the combination of molecular dynamics and NMR, sparse NMR, information that you can obtain somewhat regularly, can provide enough information at a high enough throughput to give us the right conformations which we can leverage. And so this is kind of an introduction to how we went about the methyl assignments. For anyone who's done C13 methyl labeling, you know that you can leverage C13 isotopically labeled metabolites. You simply add them into the growth media and they incorporate into the protein quite readily. And you can get really nice methyltrosine spectrum. This is an example of a C13 methyl nosy. That's a 2D projection of this spectrum, highlighting that you can get really good connectivities. And if you can do the amino acid typing, where you can determine which peaks correspond to isoleucine, and then either with selective labeling or selective dilution, figure out which peaks are corresponding to leucine valine, that really helps you in the next step, which is the automated assignment using the through space NOEs. So if you have a model of the protein or different states of the protein that you think the ligand or the apoprotein is in, or you have an AlphaFold model, you can do what is known as a pattern matching technique, where you say, these are the connectivities that we observed from the 3D nosy experiments, and we can do essentially a pattern match to the methyl network that you would expect from the PDB structure. And so, you can imagine all of the possible different permutations of NOE connectivity, and these methods essentially try to pattern match the most high confidence types of connectivities to quickly get the assignments in place. And importantly, can be leveraged to, you can put different inputs into the calculation and sort of attempt to determine which best match with the connectivity that you observed in your NOE experiments. And so that's actually really helpful to determining, both what the different states are and the local structural information. But really, in this problem, you want to think more broadly. NMR is very good at defining sort of the local atomic level resolution detail, but when in combination with SACS, and these experiments were done at UMass, you can see, get a better sense from a global perspective of the changing in conformations. And so, we have these two tool molecule states. I believe this is the apo state, and this is with the dinucleotide bound, and we can see from the radius of gyration that there's a small but significant change reflecting in the closing of the conformation of the sting protein. We can also obviously leverage the NOEs to validate and complement each approach. And then so, one of the things that we're interested in leveraging is once we have the assignments, we can do a combination approach where we do a docking to multiple states and use another second NOE-based experiment to do the intermolecular NOEs. And again, this is, maybe you just have a couple of NOEs from methyls that are nearby the ligand binding site, but it's we believe that the combination of these approaches with the molecular dynamics is sufficient to identify and guide the simulations on the correct conformation that we can then use towards for MedChem approach. So this was kind of our workflow when we were enabled with protein and NMR, but often the case that you don't have that, either the protein is really, really big, not very stable, expresses in insect cells, and it's not worth the resources to try to do the labeling, or it's very challenging. And so our focus was really to take experiments that we were performing somewhat routinely, namely the STD NMR and the HDXMS, and say, can we integrate these two in combination with computation to get at essentially where the protein is binding, or what the binding site is, if it was unknown, because crystallography was enabled, and can STD NMR actually report on where the binding mode of the ligand, a novel ligand that had an unknown binding mode. And so with STD NMR, of course, you can use it for confirmation yes/no binding, but also it provides more insights and information than that. So for example, in this case, in this figure, there's a very strong STD response for this phenyl group, and it's known or well understood that from STD NMR, you can use this approach called group epitope mapping, which essentially defines that the stronger the sort of STD response and transfer that you get, the more buried or the more the surrounding proton density is higher, whereas things that are lower are probably more solvent exposed. And so with the combination of approaches, you can imagine a workflow that we leveraged where you dock ligands where you don't know the binding site all over the protein in this sort of elaborated model, then use the STD epitope mapping to filter out binding poses that are only consistent with the experimental data, then run a molecular dynamics simulation to say, okay, they're both in agreement with the STD NMR binding pose, and they either have low RMSD or have interactions that keeps them stable in the binding pocket. And then you can also leverage HDXMS to say, okay, for example, here there's this loop that shows protection from hydrogen and deuterium exchange, and so this site and binding mode is consistent with both HDX and STD NMR. And so this is sort of an opportunity to then leverage this model as being in agreement with the available experimental data and the simulations. And the next stage would be to apply MedChem and look at the SAR and see if the SAR trends are in agreement with the model prediction. Okay, so with that, I'd like to conclude and just sort of acknowledge the team at Silicon Therapeutics and Roivant. Everybody on the list from the biophysics team to the chemistry team contributed to the work that I did at Silicon and Roivant. And obviously I'd like to thank Yasna for always giving me NMR time whenever I ask, and for her support on the facility there. And I'd like to thank everyone for your attention.